Hello, everybody. I'm very pleased to introduce Gail Schwieterman, who is an assistant professor in the School of Marine Sciences with a joint appointment in the Maine Agriculture and Forest Experiment Station. And she's going to be talking to us about climate change beneath the waves, an organismal approach. And this is her talk for affiliation with the Institute. Gail.
Thanks. Thanks, Dan.
Close the door.
So excited to be here and share some of my work with you guys. I want to start my talk by acknowledgement of some ancestral land grabs. The University of Maine was founded upon the exclusions and erasures of many individuals from the Penobscot Nation, and I have personally benefited from the displacement of these communities. And I will be discussing a bunch of work that I've done from my PhD and postdocs through my time here at University of Maine. So I'll be discussing work conducted on the unceded ancestral homelands of the Penobscot Nation, the Pamunkey tribe, the Chickahominy and Mattaponi peoples, the Chumash, the Australian Aboriginal and Torres Strait Islander peoples. And I'd like to start off with just some acknowledgements of all of the different organizations and people who have kind of gotten me to this point in my career today, specifically highlighting the School of Marine Sciences and the Darling Marine Center here at the University of Maine, as well as Dr. Erica Eliason and her lab, which was at UCSB when I was postdocing for her, as well as a suite of other places where I have done work over the past decade or so. So we're probably all familiar with the fact that the Gulf of Maine is kind of a hotspot for ocean warming. In this map, you can kind of see the colors represent sea surface temperature anomalies. So the darker the red, the warmer the anomaly is. And you can see that the Gulf of Maine is deep red. And if we were to zoom this map out, you would see that this is kind of a hotspot on a global level. But warming waters on an average scale like this are not the only thing that we need to be concerned about when we're thinking about marine climate change. We're also interested in understanding the impact of marine heat waves. And so this is something that's been picked up even by local news sources as these heat events that are happening due to changes in circulation patterns and warm air temperatures. And we can see that these marine heat waves are increasing as we move forward in time from the 1990s to the present. And temperature is kind of the hallmark of when we're thinking about global warming or climate change, but generally speaking, marine climate change is thought of as a tripart problem, with increasing temperatures, low ambient oxygen or hypoxia, and ocean acidification or an increase in dissolved CO2 concentrations. Now, these three environmental factors also impact each other. And we know that when we're thinking about the impacts on marine organisms, that temperature has all of these different effects on a suite of different physiological and biological responses. Temperature is also going to increase rates of hypoxia and can change the carbonate chemistry of the water, affecting acidification rates. And those in turn also impact a suite of biological and physiological processes in marine organisms. And so understanding these different arrows and how these different things are related is kind of the overarching, very zoomed out goal of my research program. When we're thinking about why we should care about these environmental variables, I like to argue that these environmental parameters affect physiological systems at the individual level. So we know things like temperature, salinity, dissolved oxygen affect the cardiorespiratory system, the sensory system, and the reproductive system. We also know that changes in the physiological systems at the organismal level or individual level can elicit population level responses. So changes in population growth, changes in the timing of movement, and overall survival and recruitment to fisheries that are important for the economy. Now, a lot of stock assessment scientists and resource managers are really interested in what's going on here at the population level. But I argue that in order to understand how these population metrics are changing under worsening climate change, we really need to understand the interaction between the individual and its environment. And so just to kind of contextualize things, because I know not everyone here is an organismal person or even a biologist, my work tends to fall within this fish physiology bubble. So at really fine, rapid timescales and small spatial scales. But these processes are affected by these larger scale processes that happen over broader spatial scales and broader temporal scales. So when we're thinking about aquatic organisms or animals in general, most animals are ectotherms. And I am working exclusively with marine and aquatic animals that are ectotherms. I do not work with mammals. That is Danielle's territory. And so when I say ectotherm, I mean that the internal body temperature is going to scale with the environmental temperature or the ambient temperature in that animal's habitat. And more specifically, my research is interested in the cardiorespiratory system, which involves the oxygen cascade. So these animals are experiencing temperature changes based on their environment, and that is going to affect the way that they get oxygen from the outside environment to the mitochondria, where it can be used as the final electron acceptor to create energy. And what's important is as we flow down this cascade, which is basically just a partial pressure gradient where we have high oxygen here in the water, falling down a gradient to the mitochondria, all of these steps are affected by temperature. And importantly, and as I'll mention in a minute, the heart is powering this entire process, and heart rate is also very strongly linked to temperature. And so studying the heart is a kind of central theme in my research because it is one of the first organs to fail at high temperatures. And we know based on a suite of historical studies that organismal thermal limits are going to scale with cardiac thermal limits. So understanding what's happening at the organismal level and at the organ level is important for predicting animals' responses to shifting climates. So questions, broad questions that drive or shape my lab's research are: what are the environmental conditions under which fish cease to thrive? And what factors impact an organism's capacity to respond to environmental change? And so for the rest of the talk, I'm going to kind of highlight three more specific questions or themes that have been really prominent in my lab's research program in recent years. So first, how does diet mediate responses to changing temperatures? When we're trying to understand how organisms are responding to temperature, there are a suite of factors that can be important to look at to understand that organism's response. And diet, I fully admit, is only a small portion of the suite of factors that are affecting organisms, but I think it's one that has been largely overlooked and is one that is really interesting in a time of rapid anthropogenic change. So when we're thinking about our ectotherms, we know that biological rates follow something called acute thermal performance curves. Along the x-axis here, we have temperature from a cool temperature to a warm temperature, and then biological rate on the y-axis. This can be rates of enzymes, this can be metabolism, this can be heart rate. They're all going to follow a vaguely shaped kind of theoretical model of this curve. What I want you to know is that there is a place along this curve where this is maximized. The biological rate peaks before declining as temperatures continue to increase. The rising phase of this curve is driven by thermodynamic effects on chemical reactions, but at some point, those start to break down. If an animal is exposed to warm temperatures for a set period of time, usually on the order of days to weeks, they can undergo physiological changes that allow them to acclimate. Acclimation is the process that is shifting this thermal performance curve to the right, so shifting it warmer, which allows animals then to have this optimum performance at a higher temperature than they did pre-acclimation. Acclimation can happen at a range of different levels of biological organization, changes in enzymes and the production of isozymes, changes in cell membrane composition to maintain membrane fluidity, changes in things like mitochondrial density, blood composition and hemoglobin isoforms, changes in gill surface area, and heart morphology. are all involved in this process of acclimation to warmer temperatures. So there's a lot going on in the process of acclimation. It makes sense then that diet would be important because food provides the nutrients and energy that animals need to effectively acclimate. Without these molecular building blocks and without energy, undergoing all of those biological and physiological changes is going to be really difficult for that organism. To complicate things even further, we know that climate change is causing nutritional landscapes to be more variable, right? With things like marine heat waves, we may see shifts in mobile prey items moving out of the area. We're going to see shifts in ratios of essential fatty acids being produced by plants and plankton. So the nutritional landscape is changing at a time when it's really important that animals have these nutritional building blocks that they need to acclimate to warm temperatures. And so this was kind of the basis of a study that I did while I was in Santa Barbara for my first postdoc. And we were looking at a fish called the opali, which are really unique because they're known to be a truly omnivorous fish. And omnivorous animals can change the ratio of plants to animals in their diet in response to temperature. This animal also lives in nearshore environments that are thought to be more thermally variable on tidal and diel cycles. And so what we did with these guys is we monitored their heart rate by anesthetizing them, fitting them with electrodes, and injecting them with atropine and isoproterenol to stimulate maximum heart rate, and then recorded ECGs while we heated them up at a rate of 10 degrees Celsius per hour. Now, this is not a realistic heating rate, and we are pharmacologically stimulating the heart, but this can give us a snapshot idea of the cardiac thermal tolerance of these individuals. When we heated them, we were essentially looking for them to start going arrhythmic, which can be seen here, where they would start skipping beats. And we can detect that in the ECG trace. And so these fish were fed on 2 different diet treatments at 3 different temperatures. And I'm going to show you these results now. And this was work done with PhD student Emily Hardison, who is shown here. So along the x-axis on these graphs, we have temperature at which we measured the heart rate, and then we have beats per minute on the y-axis. And so here, these fish were fed a purely carnivorous diet. So they were fed on brine shrimp only, and they were acclimated to 12 or 20 degrees. We also had fish that were fed an omnivorous diet, so they could freely choose between a local kelp species or brine shrimp. And then we had fish that were fed purely herbivorous diet, and they were only fed kelp. And what we see is that there's actually a reduction in the capacity to acclimate for the ones that were fed only the herbivorous diet. And this suggests that there are some essential nutrients that they're getting from the animal components of their diet, that if things were to shift and that animal protein was no longer available to them, it would limit their capacity for acclimation. Speaking of essential nutrients, we know from kind of foundational basic science studies on Drosophila that this can really impact that specific molecules in plant matter versus animal matter can really affect high and low temperature tolerance. And this became a concern when I moved here to Maine. So as you know, Maine is known for Maine lobster. And when I was learning about the Maine lobster fishery, something that really stuck out to me was this shift in or collapse of the herring fishery. So the lobster fishery is a trap-based fishery, and the traps are baited. Traditionally, lobstermen have used herring as the bait for their traps to attract lobsters to the pots. However, a few years ago, the herring fishery collapsed, and now it's almost impossible to get the herring for bait, and it's really, really expensive when it is available. And so this is just kind of showing through time the total herring landings, and you can see in 2019 it really dropped off and the fishery actually closed. So because of this lack of availability of herring, lobstermen turned to alternative bait items. The reason this matters is because we fish the Gulf of Maine so heavily in some regions that we can consider it basically wild farming lobster. The traps are notoriously inefficient. Lobsters are known to get in, feed on the bait, and then leave. And so by supplementing their diet with bait, we're changing the nutritional landscape that these lobsters have access to. This was work that was done with my master's student here, Madeline Young, two undergraduates, Olivia Durham and Alyssa Benoit. And what we did is we took lobsters, live lobsters at the Darling Marine Center, and we fed them on a natural diet of kelp, fish, and blue mussels to represent a diversity of things that they might be foraging on in the wild. We fed them on menhaden, which is the fish that is most similar to herring that is now available on the market. It's an oily fish about yay big. It's also called bunker or pogies. And then we also fed them salted pig hide. So pig hide is a truly novel bait. It is a terrestrial bait item. So it's going to have different compositions of essential fatty acids than marine sourced baits. And nobody is quite sure how much of this terrestrial food item that lobsters are actually consuming. So we fed the lobsters on these 3 different diets and then measured a suite of physiological parameters related to thermal performance. So this one we were measuring heart rate in the lobsters. And you can see really clearly here that there's actually no significant change from our 3 different diet items in terms of the upper thermal limits of the heart from these lobsters. And in a way, this was really exciting news, right? So negative results are not always super thrilling, but we were really happy to see this because it meant that even if these animals are eating these non-traditional diet items, it's not negatively impacting their cardiac physiology. And so by not seeing any detriments by the animals fed on the menhaden or on the pig hide, it's reassuring us that the effects that humans are having in the Gulf of Maine are not hindering the ability of these animals to respond to warm temperatures. We were also interested in looking at their metabolism of these lobsters. So this is a generalized thermal performance curve for metabolism specifically. We have temperature along the X here, and then this red line represents the standard metabolic rate, which is the rate of oxygen consumption necessary to maintain life. So this is not moving, not digesting, not investing in growth, not finding off a parasite or an infection, just maintaining life. The black line represents maximum metabolic rate. So this is the fastest that your body can possibly process oxygen and uptake it from the water. I like to say this is your I just ran a marathon metabolic rate, or I just ate a huge meal. And then the standard metabolic rate is when you are asleep and not growing and not doing anything else. The space between these two is something called aerobic scope. And basically, aerobic scope is going to be maximized at some middle temperature and be pinched off at high and low temperatures that are suboptimal. Aerobic scope, or the amount of space that you have for your aerobic scope, is how much energy you have to do all the things that make life worth living. This is where you digest meals. This is where you migrate. This is where you forage, find mates, grow, invest in reproduction. All of that stuff has to fit within your aerobic scope. And so if we integrate that area and plot it separately, we will get some generalized thermal performance curve that looks like this. And we measured aerobic scope in these lobsters that were fed 3 different diet items and acclimated to 2 different temperatures. And we will advance. And this is what we found. So we see a temperature effect where the animals held at the warm temperature were actually had higher aerobic scope compared to those held at the cold temperature, which is not what we were expecting based on the literature. But what is interesting is that at warm temperatures, we don't really see any effective diet. And so this is suggesting that even though we think that diet is really important in mediating the animal's response to temperature, at certain temperatures, whether they might be slightly above optimal, that temperature effect really overrides any temperature or any diet effects that we were seeing in this kind of short-term exposure. We also measured something called specific dynamic action, or SDA, and this is the amount that metabolism increases to digest a meal. And so here you have time along the x-axis and metabolic rate on the y, and you can see that at the point where the animal is fed a meal, you see this really large peak in metabolism, right? That's because digestion is a costly experience. Taking up proteins and assimilating, or taking up amino acids and assimilating proteins and growing, which is really what we think this curve represents, is a metabolically costly activity. And so we measured the cost of digesting these 3 different diets in lobsters at 2 temperatures. And we saw that the costs were higher for digesting menhaden than for pig hide. We did not see super strong temperature effects for this metric, but we did see diet effects. And we think that this means that the pig hide we think that the reason we're seeing this is because pig hide doesn't have very much protein in it, right? It's mostly fat and skin. It's not a lot of the meat because it's a byproduct of the pig processing industry. So if we think of SDA as how much the animal is able to grow from that meal, so how much protein is actually being assimilated into the body and being used for creation of new proteins, it makes sense that a meal that is purely fat is not going to have as much of a cost associated with it than one that is really high in protein, like the menhaden. And when we think about what does this mean for the fishery and what does this mean for the processing chain, actually having a lower SDA could be beneficial for lobsters that are entering the supply chain, because we know that lobsters are most valuable in the live market. And so if they have a lower SDA that's taking up less of their aerobic scope, they may have additional energy to devote to things like dealing with the stress of transport. So that is lobsters and diet. But I kind of want to leave you with this diet theme, just thinking about how climate change is shifting nutritional landscapes. So even though the lobster example was, a bit of an artificial example of how diet might be changing, we know in general, thinking broadly, that climate change is shifting food availability, particularly before and after marine heat waves. We know from studies outside of the Gulf of Maine that this can reduce diversity and availability. We also know that climate change is changing food options. So marine heat waves are causing drops in diversity of organic matter, of plant matter, and of species richness. And we're also seeing changes in food quality. So even if the species diversity doesn't change, the actual nutritional quality of that diet, due to shifts in essential fatty acid composition, is changing. And so we're seeing lower ratios of DHA and EPA compared to other fatty acids that are known to be essential for many marine organisms and for humans. So thinking about how diet is shifting and then how that's impacting organisms and mediating their ability to acclimate to changes in their environment is one theme of my research program. Another broad theme is thinking about how oxygen restriction impacts upper thermal limits. So I mentioned that one of the kind of 3-pronged issues with marine climate change is hypoxia, or oxygen limitation. And we know that as ectotherms get too hot, that we start to see this decline in performance, right? So again, my generalized thermal performance curve, we see this drop when animals get too hot. And we know that if animals are too warm for too long, that they will die. What we don't fully understand, despite, decades and decades of animal physiology research, is what is the mechanism causing this decline and driving mortality at high temperatures. And I am of the opinion that it is related to oxygen availability. So going back to our metabolic rate thermal performance curve, if we think about an oxygen-limited environment, we would expect this curve to change by dropping down that maximum metabolic rate line, pushing down and constraining aerobic scope from the top. What this is going to do is it's also going to shift the point at which those two lines pinch together, which we consider a critical point, which can also be a mortality point. It's going to shift that left or to a cooler temperature. So under oxygen limitation, we would expect thermal limits to be lower. And we can actually measure this. So this is a description of this method or of this metric called critical oxygen pressure, where we basically have an animal in a chamber and are measuring its metabolic rate while we drop the oxygen in that chamber using compressed nitrogen. And we look for this inflection point where they're no longer able to maintain their standard metabolic rate. So if the animal spends too much time down here below this inflection point, they will die because they're not able to maintain that like just staying alive metabolic rate. They're not able to do maintenance on their bodies. And what this means is when we shift this inflection point lower, that animal is more hypoxia tolerant. And if we shift it to the right, that animal is less hypoxia tolerant. And so I measured this in 3 different species from the mid-Atlantic, clear nose skate, summer flounder, and then thorny skate, which thorny skate also are up here in the Gulf of Maine. And I measured them at several different temperatures and at 2 different pH levels. And the y-axis here is going to be the critical oxygen pressure. And so what we're looking at here is the lower this is, the more hypoxia tolerant these animals are, or the better they're going to do under low oxygen conditions. And so what we see is that looking at the 2 different pH levels, when the animal is exposed to a low pH environment, so 7.4 is quite a bit lower than we would expect to see in estuaries where these guys live, we see a higher partial pressure or for a critical point, meaning that this animal is less hypoxia tolerant when they are also dealing with the co-stressor of simulated ocean acidification. We also see the same pattern in summer flounder and in thorny skate, specifically looking at the ambient temperature here. So under ambient conditions or mild warming for the clear nose, we see this effect of this pH treatment of this co-stressor. But we do not see any significant changes in the critical oxygen saturation or partial pressure when we get to warm temperatures. And so this is suggesting that at warm temperatures, we're overriding the effect of that pH, and we're getting in this interaction where temperatures and oxygen are becoming codependent, and oxygen limitation may actually be driving upper thermal limits. That was really exciting, but then I went to French Polynesia and we did a similar test on blacktip sharks and exposed them to a suite of different multi-stressor scenarios and found no difference in their hypoxia tolerance. So this idea that the co-occurrence of stressors was really driving the ability of animals to withstand hypoxia and to have high thermal limits was kind of fell apart when we added in this tropical species. Caveat is that we did use a different metric. We used the onset of muscle spasms rather than the inability to maintain standard metabolic rate. And that this species is a lot more active than the 3 species that I measured on the previous slide, and that something about activity level may be mediating these responses to co-stressors. But what I think is really interesting is in my work alone, I have seen have examples of additive, synergistic, antagonistic, and masking effects when we're thinking about these co-stressors and how animals are transporting oxygen. And so I think this is really interesting and made me want to delve even more into the mechanisms underpinning these different responses to multiple concurrent stressors when we're thinking about oxygen transport. And so my favorite oxygen cascade, we decided to take all of that work at the organismal level and try to zoom in on things that were happening at the suborganismal level. And so using the method I already described, where we were measuring ECGs in the heart, I did another experiment while I was at UCSB on opali, and we conducted that same test where we were rapidly warming these fish under 3 different oxygen scenarios. So one was 100% oxygen saturation. One was super saturated, so it was 200% saturation, and then one was hypoxic at 20%. And we found that when we artificially limited oxygen, the fish had lower cardiac thermal tolerance. However, when we artificially oversupplied the fish with oxygen, it had no change compared to normoxia. So oxygen can be the limiting factor in driving cardiac thermal limits, but it is not always the limiting factor. And so there's something else going on at high temperatures which cause those arrhythmias. I'm guessing it's something to do with calcium cycling and calcium leak, but this is suggesting that in scenarios where oxygen is limited, it is going to negatively impact thermal tolerance. What am I doing on time? I've also in my lab been interested in other kind of suborganismal ways to look at oxygen transport. And so specifically interested in hemoglobin. So the way that we assess hemoglobin oxygen affinity is using these curves where we have oxygen concentration on the x-axis and the amount of oxygen that is bound up to hemoglobin on the y-axis. And it typically follows this kind of sigmoidal curve. 100% saturation is not super informative because no matter what isoform of hemoglobin you're producing, it will eventually get 100% saturated. So instead, we use this 50% saturation point, which we can then look at if we drop straight down to the concentration, and that concentration is what we call P50. And we know that there are a suite of different factors that can affect the P50 value, primarily temperature and pH, but also the isoform of hemoglobin that's being expressed or the ratio of different isoforms. And then there are a bunch of allosteric modifiers that can affect this protein's ability to bind up oxygen as well. And so depending on the species that we're working with, we would expect the shape of this curve to change in different ways, depending if we're looking at different temperatures or if we're looking at venous versus arterial blood. And so this is something that my other master student, Emma Smith, is working on in the Little Skate. So these guys are native to the Gulf of Maine, and they're unique because they lay eggs where the embryo will develop for up to 12 months, depending on temperature. And this means that the embryo is tethered to the benthos and cannot move during development if conditions become unfavorable, if there's a marine heat wave or if there's on a hypoxic zone. And so what Emma did was acclimated embryos to 3 different temperatures and 2 different oxygen concentrations, and then measured development in a bunch of different ways. And she is currently processing blood that we took from hatched embryos as well as from the parent population to compare oxygen tensions in P50s to see if being reared or developing under these different conditions is actually shifting the oxygen affinity of the blood. And this work is still ongoing, but just a little bit of a teaser. This is what they look like when they hatch. They're super cute. And we did see effects of rearing temperature on development time or time to hatch. So more results coming with that project. But I think that looking at this kind of suborganismal level is going to be really interesting and really informative to understand how this species is responding to marine climate change. And when we're thinking about understanding or looking for these suborganismal mechanisms, I get this question a lot in public talks is how are we going to predict the winners and losers of climate change, right? We know some animals are going to fare better than others, and realistically, it's not feasible financially or logistically to go out and measure the physiology of every population and every species. But I'm hoping that having these mechanistic understandings or to find trends in activity level and species and phylogeny of which animals have the physiological capacity to respond to things like low oxygen and warm temperatures can help us narrow down and kind of make better guesses about who's going to be a winner and who's going to be a loser. Okay, so next is thinking about environmental variability and how this is going to shape phenotypic plasticity. So when we're thinking about the natural world, we know that variability is everywhere all the time, always. And so these are some factors that are important for aquatic and marine species that we know are going to vary over spatial and temporal scales. Now, traditionally in my field of experimental animal physiology, lab-based experiments tend to use mean values. And this little animation is just showing that relying solely on consistent means is not representative of what's going on actually in the environment and the kind of variation that animals might be experiencing in the wild. Right. So just an example from my time in Santa Barbara, this is logger data that I had placed in prime opali habitat near Stearns Wharf. And this is the mean of this temperature trace. And you can see that it's really not capturing the variability that we're seeing on seasonal scales, nor the variability that we see on tidal and diel scales. And so when we're thinking about communicating our results and kind of making our science applicable to broader audiences, comments that I've gotten personally when I've given talks about experiments that I've done are I think totally valid in being critical, saying, so I've had feedback that my experimental design is an oversimplification, that the results that I'm finding in the lab don't match what people are actually observing out in the wild when people are on the water. And then questions about, well, even like the temperature treatments that you use seem really extreme. Don't these animals have thermal refugia? Or aren't they able to move around and escape these kind of really intense warming scenarios? And I think that all of these comments are completely valid, right? And so something I've been thinking a lot about recently and kind of challenging people in my field to think about is, can we be more than the mean and should we be more than the mean? And so there are arguments to not incorporate environmental variability into our research, right? Environmental realism is logistically difficult. It's really hard to choose which factors matter and to make them vary at these scales, the spatial and temporal scales that are relevant to animals in a lab setting. It also can obscure the results. So when we have really clean, defined experimental designs, it can be easier to elucidate mechanisms and derive causative factors of the results that we're seeing. However, if we invest in creating environmental realism in the lab, it will be easier to directly link results to real-world scenarios. And it can help illuminate synergistic and antagonistic processes that we might miss by only looking at these kind of static treatment levels. And so when we're thinking about variability, we can't not talk about Jensen's inequality, which is a mathematical concept that has kind of been co-opted by physiologists when we're thinking about should we or should we not incorporate variability into our treatments. But in lay terms, it just says performance is going to depend strongly on the variance in skewness in temperature. And we can kind of walk through what it means graphically. So In this scenario, we have temperature on the x-axis and some metric of performance, let's say metabolism on the y. And we have two different populations, one that is acclimated to a stable temperature here in the mean in the blue, and then one that's acclimated to a variable scenario with the same mean temperature. So both these populations have the same mean temperature that they are acclimated to. For the static population, we can expect that they will be aligned, their thermal performance curve will be aligned with their acclimation temperature, right? So they are experiencing this consistent temperature. They would undergo acclimation and physiological changes to maximize their performance at that temperature. If we are trying to predict what the performance will be from individuals acclimated to the variable treatment, we can say, okay, we know that they're going from plus or minus 3 degrees from that central temperature. Why don't we just plot that on this graph and draw a line? And Jensen's is going to say that we predict that they will have a lower performance at that mean temperature compared to individuals that were in the static acclimation treatment. The problem with this is this doesn't account for the fact that thermal performance curves are plastic. So we know when animals are held under any temperature treatment, whether it's consistent or stochastic or whatever, that they have the capacity to change the shape and breadth of that thermal performance curve, things like the height and the upper thermal limits. And so without fully measuring a full performance curve for organisms exposed to static versus variable conditions, it's impossible to predict how that performance might vary between those two groups. And so work that I did in California on our trusty opali, I was interested in kind of exploring this question of like, does variability matter? And if it does, when does it matter? And so these are temperatures from the habitat near Stearns Wharf where we collected these animals. And so you can see that there is variability seasonally, but also kind of within seasons. And this is an area that's also subject to marine heat waves. So this was the experimental design. I had 3 static temperature treatments and then a variable with the same mean as 16, and then another variable with a mean at 19 degrees. And we did our method of measuring cardiac function to determine upper thermal limits in this species. Here, I'm not going to be showing the data for the point where they become arrhythmic, which is here. I'm going to be showing the data for the Arrhenius breakpoint, which you can calculate by feeding this data into R and then doing some modeling and plotting this in Arrhenius space. But here are the results. So the acclimation treatments are along the x-axis and temperature is along the y. And what I'd like to highlight here is that there were no significant differences between treatments that had the same mean value. There's a trend of them trending high when they were in the variable treatment compared to static, but there was no statistical significance between either of these pairs. And so for this species, looking at cardiac thermal tolerance, variability maybe doesn't matter. And we know that this is a rocky intertidal species, that they are going to experience high levels of thermal variation on really fine time scales, tidal scales, diel scales, seasonal scales. And so maybe for a species like this, it doesn't really matter if we incorporate thermal variability into their acclimations in the lab because they're just going to have broad thermal performance curves regardless, right? Like maybe this species doesn't have the capacity to really narrow that thermal performance curve and become a specialist. So that was like, great, then all the other experiments that I did prior to this with this species are totally valid. But I think it's really like a lot of physiology is something that can be really population and species specific. So continuing in this kind of theme of thinking about variability, I did a project this past summer here at Maine using Atlantic salmon. So Atlantic salmon are culturally and commercially valuable, and all sea-run populations were extirpated except for Maine's. So we have 6 DPS, distinct population segments in Maine that are genetically unique and endangered, and I did not touch those because they are endangered and I didn't have the permits for that. So instead we used hatchery fish from a hatchery here near Orono, and then hatchery fish from Connecticut. And those fish were originally from the Penobscot strain, but have been in Connecticut for over 2 decades. So we assume that they are also genetically distinct. And this work was done in collaboration with Dr. John Kosick from NOAA, and then Lucy Henthorne, who was a NOAA Hollings Fellow, and Bryn Cleary, who's an undergrad in the School of Marine Sciences. And this is data from the Penobscot River that's showing kind of the variability that we see during the summer. And we chose these temperatures to represent our static and our variable treatments. So our static treatment was held at 22.5 consistently, and then our variable oscillated from 20 to 25 every day. And we acclimated fry from these 2 different populations to these 2 different thermal treatments for at least 3 weeks, and then measured something called critical thermal maximum. And it's basically a test where you heat the fish up really, really quickly, like 0.3 degrees a minute, and wait for them to lose equilibrium and flip over. It's a repeatable test. It's a non-lethal test. And it's really useful in comparing different populations or acclimation treatments really quickly in a non-lethal, non-invasive way. And so these are the data. We can see that the 2 groups that were acclimated to the variable conditions had warmer CT maxes, so they had higher upper thermal limits compared to the ones that were acclimated to the static condition. But what I think is really cool, and we didn't think to do this until the end of the experiment, was I convinced my undergrad students that we should measure CT max in the middle of the night, right? So the fish that were in the variable treatment always were warm during the day and cool at night. And we were running these tests during the day because that's when we were in the lab. But I convinced them that we should come in the middle of the night and do a CT max test. And that was where these guys fell out. So these guys had a higher thermal maximum than the static ones, even though in the middle of the night, they were held 2.5 degrees less than the ones that were at the static treatment. And so this suggests that acclimation to variable environments is going to broaden that thermal performance curve. And even in periods when they are not warm, they are going to maintain that ability to survive at higher temperatures. And so we're also seeing that it is a little bit lower than when they are tested during the day. This suggests that these animals are adjusting their thermal physiology on the order of hours, right? If they were just acclimated and fully acclimated and not changing, we would expect that the warm and cold ones, or the ones during the day and during the night, should have the exact same CT max, but that's not what we saw. And so this is really exciting. It means that these early life stage fish that are considered to be highly vulnerable to temperature stress actually have the physiological mechanisms to alter their physiology really, really quickly, like way quicker than we thought. Now, the caveat here is, of course, as they were acclimated to a very prescribed thermal regimen that is not going to be the same as out in the rivers where there's a lot of stochastic variation. But I still think this is really, really exciting. And this actually mirrors work that I did in my second postdoc when I was at Carleton College, where I had undergraduates working with Fundulus heteroclitus or mummy chogs. And I was smarter back then, and we acclimated fish to a reverse diel thermal cycle, which meant that they were warm at night and cold during the day. And so for this one, we could just come in and measure CT max during the day. We didn't have to come in the middle of the night. And again, we're seeing that there's a difference between these diel and reverse diel temperatures, suggesting that these animals are also changing their thermal physiology on the order of hours. And so this is really excited. This is definitely something that I am eager to dig into more in the mechanisms and try to understand what is driving these patterns. But shameless plug for thinking about variability in physiology. A couple of years ago, I put together a special session at the Society of Experimental Biology with Warren Berggren, Chris Wood, and Emily Hardison. And next month, our special issue in Philosophical Transactions of the Royal Society is coming out. That was based on that conversations that happened in that session. And I have 2 papers coming out in that special issue if you are interested in thinking more about variability. The first is kind of a historical perspective summarizing the state of variability research up to now. And then the second is meant to be kind of a how-to guide for people who are interested in thinking about incorporating variability into lab-based experiments. So, okay, so to kind of wrap things up and bring things home, I mentioned at the beginning of the talk that marine climate change is a tripart problem with increasing temperatures, low ambient oxygen, and ocean acidification. I hope that I've done a little bit of work to convince you guys that I don't think that's actually true. I think that marine climate change is a suite of problems that we don't fully understand. And while it's definitely important to understand the impacts of temperature or hypoxia or ocean acidification, looking at those in isolation is not enough. We also need to be considering the timing of things, shifts in species distribution, shifts in diet, species interactions, all of these other things that are impacting not only behavior and population size, but also physiology. So when we're thinking about marine climate change, we know that co-occurring stressors elicit multiple responses at multiple levels of biological organization, and that changing environments are complex on spatial and temporal scales. And understanding how individual populations and species are responding can help elucidate mechanisms underpinning resilience. And that's really kind of the overarching goal that I definitely can get lost in the weeds and get too excited about the physiology. But the overarching goal of my research program is to understand mechanisms underpinning vulnerability and resilience to changing habitats in the marine realm. And with that, I would be happy to take any questions.
We do have a few minutes for questions. I have one.
Yeah.
So you do experimental work in the lab. I'm wondering but you talked about the importance of actual natural variability. Is there any way to do any of this work out in the real world, and do people do that? And for my own particular interest, I even do it tracking the effects of El Nino along the coast of Peru or other similar areas where you have these aperiodic warmings.
Yes. So there are people who do what we call like stream side physiology, where they will take the equipment out into the field and they will do measures of metabolism and blood chemistry, like on a boat, like they'll catch a fish and shove it in a respirometer, or they'll do it stream side where they'll catch the fish and they will basically be drawing ambient water in as like the temperature bath for that experiment. And so it can be done. It is an immense amount of work, and it is you're taking instruments that are, tens of thousands of dollars that are not designed to be used outside of the lab into the lab setting. So there's always some nerves associated with that. But it is something that people are doing. The data are really messy and they're hard to kind of reconcile. But that is something that is happening. Joe Zadlewski has a a swimming respirometer that the idea was to take it streamside and look at salmon, but that hasn't gotten off the ground yet. In terms of doing it in Peru or Ecuador and kind of looking at El Nino stuff, I'm not personally aware of anyone who's doing that. I know that the Charles Darwin Station in the Galapagos has done a little bit of kind of like tracking real-time responses and thermal tolerance to the organisms that live there. But most of their work is more survey-based rather than like experimental-based. Yeah.
Thank you.
Mm-hmm.
You talked about so many interesting, really fascinating things here. I can't even begin to decide where to ask the question, but one of the things that I'm a plant person and I study long-term plant responses to climate change during ice age periods of time. And so we've been very interested in assessing some of these same things for what it means when plants populations change and so forth, distribution, abundance, and that sort of thing. And one of the things that keeps popping up as a possibility, although hard to identify for sure, is the extreme events. You've done a really neat job of assessing different conditions during that are at least tolerable to the species, but sometimes extreme events can be very brief, but perhaps dramatic for a population or at least for individuals. And is that, your measurements help identify those things?
Yeah, so they can. I think like scale is always like something that's hard to kind of figure out, like where are you trying to situate your experiment? And so like some of the work that I showed when we were trying to assess the impacts of marine heat waves, we were picking like, okay, in the past 5 years, what's been like the warmest temperature these organisms experience? But even that, I don't know would be if it's really considered like an extreme event, especially given the frequency that they're happening right now. I will say so. One, aquatic environments tend to have lower variation than terrestrial environments due to water's high heat capacity. I will also say that when I'm thinking about extreme events, I would almost think less about temperature and more about oxygen. So we know like the Gulf of Mexico is really well documented that they have these dead zones that occur that are actually completely anoxic water. And so those are pretty well studied and are known to have really detrimental effects on organisms that cannot move out of them. So you get these large fish kills, and changes in have these cascading impacts of like fishers have to go further out offshore and whatever. But yeah, I think oxygen might be a more a better analogy to that kind of thing, because those are also really brief. So dead zones can last on the order of days, sometimes months, but usually days, depending on the size and the location and the level of water mixing that you get.
I have another question, but go ahead.
The chemistry of the Gulf of Maine is changing, partly from dilution by different ocean currents bringing in, so on, various constituents, but also the landscape is contributing a huge amount now of calcium and magnesium and potassium and phosphorus, and I'm wondering if that's a concern that you have in the Gulf of Maine.
It's not something that I've spent a lot of time thinking about. And if I had to hazard a guess, it would be, I would be more worried about invertebrates than vertebrates with those kinds of changes in water chemistry.
The invertebrates are part of the food chain, of course.
Yeah, definitely. Yeah. It would definitely be something to look more into. I'm not super familiar with it at this point.
I had a question about, I was really struck by the curves you kept showing that were so asymmetric with it you know, the effective temperature being gradual, and then it just was like abrupt.
Mhm.
Are there similar effects for things going the other direction, getting colder? Like, is cold tolerance? Probably no one studies cold tolerance.
Oh no, people, yeah,
Is
people
there a
definitely
sim-
study cold tolerance, yeah.
Is there a similar effect, or is it better equipped to deal with colder temperatures?
Yeah, so I would say generally the shape of the curve is going to be more gradual at the cold tolerance end. It is obviously going to change depending on the metric and the species and the population. There is a lot of work on cold tolerance. You know, when we're thinking about marine climate change and we're thinking about poleward expansion of species, that's a big area of research, is understanding, okay, what's the farthest north or south that they can go? And that's often limited by cold tolerance. And so there's yeah, that's definitely like an area of research. It is also something that people are looking for, like Arctic char and other like true cold water species, is if that cold tolerance is plastic at all, or if it's that they actually need to be that cold. And like if they warm acclimate, do they lose that cold tolerance, and is that going to be detrimental during like winter seasons? So it's not something that I personally have done at this point, but it's definitely like a big area of research in my field.
One of your friends that you probably Brenda knows that she works in that area, Bruce Seidel, who is
Yeah.
getting fishes and increase.
Yeah.
Yeah.
That's a great example.
Yeah.
I'd like to go back to extreme events, and I was thinking as you were talking about a couple of particular ones. One, for example, in the last 10 days or so, the southeast coast has been extremely cold. I mean, really, really unusual for you. And those coastal littoral waters down there must have been shocked completely. And I mean, my guessing one of the whole populations would have been probably destroyed or affected by that. Or those are many, many tens of degrees below what they're used to.
Yeah. So I will say, so again, not having done a whole lot on the cold tolerance side. So if animals are mobile, so if they're living like outside of the intertidal and are mobile, they would probably leave. If they are in the intertidal or not able to leave, they may have perished. They may not have. So we know that particularly for invertebrates that live in the intertidal is one of the most stressful environments that you can possibly live in. You get huge variations in salinity and air exposure and temperature and all this stuff. And so those organisms tend to be extremely hardy and extremely resilient to environmental stress. There are obviously limits and bounds to that. So I can't say for sure, one way or another, but
Well,
yeah.
I was thinking, for example, about those gigantic salt marshes down there that are breeding ground for a lot of fish species, if I
Yeah.
understand them right. And probably it was difficult for those animals living there to move there, to move far enough. And it must have been very cold in those places.
Yeah.
For a period of time. Another example that's interesting to me is the salmon one, because I've actually worked on salmon over longer periods of time. One of our colleagues in ecology and archaeology and I have done some work there, and it turns out that there are sea-run Atlantic salmon bones in some archaeological sites. And they occur only at times in the past when conditions were relatively cool and there was a lot of water in the rivers. But at the times when the rivers became much warmer and lower flow, which happens occasionally here,
Mhm.
I mean, they get really warm. when
Yeah,
the flow
salmon
is
don't
low
like
and
that.
dry in summer, so of course the salmon aren't there. But
Yeah.
it is interesting to think about those changes through time. The species did apparently come back in when conditions were appropriate, and then probably weren't here for a while too. But really, as you say, the mobility is a big part of the story here.
Yeah, and I mean, I didn't even like touch on like transgenerational effects and like, there is evidence that temperatures that the parents are exposed to through epigenetics can then impact the thermal physiology of the offspring. So there's lots of nuance there.
Thank you very much. I've got to run.
Yeah, thank you guys.